So, how does it all work? How are traits passed down from one generation to the next? It's one of those huge fundamental questions in all of biology, really. We're going to dive into the story of inheritance, which is the absolute basis of heredity. And let me tell you, it's a story that unlocks the secrets of life itself. It really all starts with a question that sounds so simple, but it's actually incredibly profound. You probably thought about it. Why does an elephant always have a baby elephant? Why does a mango seed only ever grow into a mango plant? And think about it, are those copies perfect? I mean, why do some siblings look almost identical while others look so different? That's the big puzzle we're going to try and solve today. Okay, so to even begin solving this puzzle, we've got to get our heads around two key ideas, two forces, really. You've got one force that creates sameness, continuity, and then you have another force that creates all the differences, the diversity. These two concepts, they're the foundation for everything we're about to explore. All right, first up, inheritance. This is that first force, the one that's all about continuity. It's the process that makes sure characters get passed on from parent to child. It's exactly why that baby elephant is always an elephant. This is the absolute bedrock of heredity. But here's where it gets really interesting. Inheritance has this amazing partner, or counterpart, called variation. This is all about the differences, right? It's the degree to which kids differ from their parents. It's what makes every single one of us unique, and it's basically the raw material for all the mind-blowing diversity we see out there. These two things together, inheritance and variation, they literally write the story of life. Now, for centuries, people kind of knew this stuff, right? They had a gut feeling about it. Farmers knew to breed the best crops, the best livestock, but the actual science behind it, total mystery. That is, until a monk, Gregor Mendel, started doing some incredible experiments in, of all places, a quiet monastery garden. So for 7 straight years, this guy Mendel just meticulously, carefully crossbred pea plants. He figured it all out and published his work way back in 1865. But nobody got it. The world just wasn't ready. His work, the very foundation of modern genetics, basically just sat there, completely unnoticed for 35 years. Can you imagine? Finally, in 1900, it was rediscovered, and everyone suddenly realized what a genius he was. So what exactly did he do that was so revolutionary? Well, let's walk through one of his classic experiments. It's beautiful in its simplicity. He took a true breeding tall pea plant and crossed it with a true breeding dwarf one. Okay, so in that first generation, we call it the F1 generation, every single one of the new plants was tall. Every single one. It was like the dwarf trait just vanished. Poof! But then, and this is the brilliant part, he let those new tall plants self-pollinate. And guess what happened in the next generation, the F2? The dwarf trait popped right back up. That was the light bulb moment. The trait wasn't gone, it was just hiding. So to make sense of this, we can use this amazing little tool called a Punnett square. It's basically a simple grid that helps us visualize the probabilities. It shows us all the possible ways that different versions of the height gene, what we call alleles, can combine from each parent. And look, you can see you get one combination for a pure tall, or TT, 2 combinations for a hybrid tall, TT, and 1 for a pure dwarf, TT. And this simple square perfectly explains what Mendel was seeing in his garden over and over again. When he counted up all those F2 plants, he always found pretty much the same thing. About 75% of them were tall, and 25% were dwarf, a perfect 3-to-1 ratio. This wasn't a fluke, a predictable mathematical pattern in biology. That was huge. It meant there had to be some kind of rules, some underlying mechanism at work. So based on this rock-solid evidence, Mendel came up with a truly revolutionary idea. He proposed that traits are controlled by these little separate units he called factors. Today we call them genes, and he said they come in pairs with different versions that we call alleles. And here's the kicker. In that pair, one allele can be dominant, it shows up, while the other is recessive, it gets hidden. This idea perfectly explained why that dwarf trait seemed to disappear and then pop right back up. Okay, and this brings us to a super important distinction. You have the genotype, which is the actual genetic makeup, the code itself, like TT or TT or TT. Think of it like the recipe. Then you have the phenotype. That's the physical trait you can actually see, like being tall or dwarf. That's the cake you bake from the recipe. And as we just saw, you can have two different genotypes, TT and TT, but they both give you the exact same tall phenotype. From these incredibly elegant experiments with peas, Mendel came up with his foundational laws. The law of dominance? That explains why all those first-generation plants were tall. And the law of segregation? That explains that the two alleles for a trait actually separate when making reproductive cells. Now, the really crucial thing to get here is that these weren't just observations about peas anymore. For the first time ever, these laws turned the study of heredity into a predictive science. That was a game changer. So, Mendel's laws gave us this incredible, solid framework. The foundation was set. But as scientists started digging deeper, they found out that the story of inheritance, well, it had some pretty fascinating plot twists. Okay, plot twist number 1, incomplete dominance. So with Mendel's peas, the tall allele completely masked the dwarf one, right? It was all or nothing. But check this out. With snapdragon flowers, if you cross a red one with a white one, you don't get red or white. You get pink. The new phenotype is a perfect blend right in the middle of the two parents. How cool is that? But wait, it gets even more complex. There's also co-dominance. Now, in this case, instead of blending, both alleles show up and express themselves, fully and separately. The classic example is our own ABO blood types. If you get an A allele from one parent and a B from the other, you don't get some kind of blended blood type. Nope, you get AB blood, where both the A and B markers are fully present on your red blood cells. They're co-dominant. So think about it. Mendel had described these factors, these genes, but they were totally abstract ideas. For decades, nobody had a clue what they were physically made of or where you'd even find them inside a cell. Solving that mystery, well, that's the next chapter in our story. Yeah, this quote really gets to the heart of the problem facing the next wave of scientists. I mean, Mendel's math was airtight, it was convincing, but biologists needed to see it. They needed physical proof. Where in the world were these factors hiding? Well, right around the turn of the 20th century, microscopes got a whole lot better. And suddenly, scientists could see these little structures inside the nucleus of cells. They called them chromosomes, which literally just means colored bodies because they soaked up the dye they used for staining. And the really crucial thing was they watched them do this very precise little dance. They would double and then divide right before the cell itself divided. And here it is, the giant aha moment. A couple of scientists, Walter Sutton and Theodore Boveri, noticed something incredible. The way these chromosomes were behaving during cell division was a perfect mirror image of how Mendel's abstract genes were supposed to behave. Think about it. Genes come in pairs, so do chromosomes. Genes segregate into different cells, so do chromosomes. It was an absolutely astonishing parallel. They had found them. The chromosomes had to be the physical things carrying Mendel's factors. I mean, what a discovery. This connected Mendel's abstract rules right to the physical machinery inside our very own cells. And that connection, it has absolutely profound implications for us, especially when it comes to understanding our own health and the diseases that affect us. To really see just how powerful this connection is, we just have to look at one example: sickle cell anemia. It's a story that starts on the tiniest scale imaginable. It all boils down to this number: 1. It starts with just one single error, a mutation in just one single base pair in our DNA. So here's exactly what's going on at the molecular level. That single base change, from GAG to GUG in the gene that codes for hemoglobin, well, it causes the cell to slot in the wrong amino acid. Instead of glutamic acid, it puts in valine. That's it. It is literally like a single typo in a giant 1,000-page instruction manual. But the consequences of that one single typo are just staggering. Because of that one wrong amino acid, the hemoglobin protein is altered, and that causes red blood cells, when oxygen is low, to twist from their normal healthy disc shape into a stiff sickle shape. This one tiny molecular change leads to a lifetime of complex and serious health issues. It's a powerful and kind of scary demonstration of the immense power packed into every single one of our genes. And that really brings us to our final thought for today. Think about the journey we just took, from a monk's quiet garden all the way down to the very code of life itself. We've seen how a few simple rules can lead to all this incredible complexity, and how the tiniest change can have the most massive impact. It just leaves you wondering, doesn't it? If this is the story of just one tiny change in one gene, what other incredible stories are just waiting there to be read in our DNA?